Good afternoon, everybody, and thank you for being here. If I may just continue the conversation or presentation we just heard, patients are waiting, and they're not just waiting, they are eagerly awaiting their cure. And I think before starting into the presentation, I think we are all here in imagining that we treat a type 1 diabetic patient or treat a fibrotic patient to provide them with a cure. And this is essentially what have been driving us to build up an integrated platform delivering iPSC-based cell therapies to clinical testing. So we are exclusively focused on autologous approaches, allogeneic approaches in iPSC, meaning there will be, in our belief, still a place for autologous treatments because they do what they do and they're doing it quite well. But on the other hand side, moving forward, we took the decision to really focusing on allogeneic iPSC-derived products, and we have positioned ourselves as one of the key players who want to support partners in all ideas that come from inception up to the GMP manufacturing for phase 1 and phase 2 clinical supply. So and this is only a nutshell, I don't want to present Evotec because that's boring for everybody. We are about 4,000 scientists working with partners. We are creating pipeline assets with partners, supporting them in their research activities and development activities. Generally, we think beyond FTE, so we are always not geared towards just delivering. We are also looking for collaborating with our partners. We have different partnership models and a strong track record in building capital-efficient execution, and we are operating across the planet, numerous facilities for development and research activities. So Evotec cell therapy, as I said, it's iPSC focused. We have built this because we have shown in the past in the whole Evotec universe that we can de-risk development programs. So we early on ask ourselves, what does it take to move cell therapies fast and efficiently towards IND stage? And we believe there are 3 core pillars that we are always following. On the first important piece, this needs to be integrated. So iPSC development and the development of a candidate does not, how can I say, work standalone. You always need the integration into a therapeutic area knowledge as well as a professional environment to work together with our partners. That's what we call integration, and that's what we are geared towards. This integrated approach, although sounding costly, you have therapy, you have technology, you have management, needs to be sustainable. So meaning we are always looking for to be competitive with our peers and to provide very cost-efficient solution. And we are doing this always in a qualitative approach. So we know for more than 30 years how to drive projects from early ideation up to IND. We are doing about 20 IND filings for our customers per year. So we really know what it means to deliver and to retain flexible and efficient to provide the outcome. With this idea in mind, we have built a platform that we call end-to-end platform for iPSC-based therapies. And end-to-end for us means you can start with us as early as exploratory. So we can run iPSC differentiation, we can do gene editing, we can run in vitro proof of concept, and you can continue the journey with us up to the clinical stage, meaning we would be handing over the full IND package to you, working with you on the IND package, and also perform the clinical supply. And as I said, the middle ground is really important to integrate from very early on the pathway towards clinic into the whole research plan, meaning the preclinical research and preclinical development is key for this. In the meantime, we are deploying more than 100 scientists and cell therapists in our facilities in Göttingen in Germany and here in Italy in Modena. I truly believe we have a leading iPSC expertise, and that stems from the availability of iPSC lines within GMP-grade quality. It stems from our experience to develop GMP-compatible differentiation protocols, which is often underestimated in terms of complexity and availability of materials. We have, as everybody would assume, the needed GMP-compatible gene editing technologies available. But it's not only generic technologies that we have in-house, we also work with proprietary hypoimmune technology and proprietary kill switches. So overall, this coupled with GMP bioreactors, upscaling, process development, gives us a very, very unique position in the early preclinical stage to collaborate with partners and to develop our own proprietary programs for partnering. Very clearly, we are not a company that is developing our own projects for clinical development, so clinical development is always done in sponsored by our partners. A very integral part of this journey is what we call our cell factory. So Modena is a little bit north of here, south of Verona, and in this facility we are really closing the gap of the current iPSC state of development, where not a lot of projects have been actually entering clinical development, so there is still a huge learning to come from a research process development process to a fully compatible GMP clinical manufacturing. So we have built out this facility to really bridge from the development into clinical application. This facility has been audited recently by IFAR, and what makes it unique, this facility is approved for autologous, allogeneic, and GMP gene editing, which is quite unique in its setup. Whereas we are also running some autologous programs in this facility, our own focus is really to use it for iPSC-derived therapies and gene editing. The facility is very flexible, so we deploy 5 clean rooms, BNC status in this facility, and they can be utilized for gene editing and also for different iPSC workflows. And this is coupled with a large area for process development, because this is where today iPSC therapeutics are mainly developed. It's in a preclinical stage that will require moving forward process development activities. What we are known for anyhow is, with our many years of experience within Evotec as a company, is our regulatory affairs and also clinical translation expertise. And this is not only embedded in Modena, but also with the colleagues who are running these departments from our site in Verona. So overall, we are really built to support and develop our own projects to deliver the next generation off-the-shelf products. And I think everybody in this room knows this workflow, so I'm not really going into every detail of it. The uniqueness of iPSC is that you really are developing your GMP master cell line, either edited or non-edited. And again, all that you're seeing here around this circle is in-house. It's GMP lines, it's gene editing, engineering, manufacturing, off-the-shelf frozen products. And what I would like to point out essentially are those little blue boxes where we feel that a very strong emphasis needs to be taken to ensure the quality. And what we often see when we look into protocols and have discussions with potential partners, those are areas where we feel that not enough emphasis often is given. For example, when you look into the gene editing, in particular, the genetic engineering needs to be confirmed that you have still an integral and intact cell line. So a full genetic QC is mandatory, and this does not only imply you're using, karyotyping or 1 or 2 assays. We are using a set of up to 10 different types of QCing genetic integrity. This is important simply to ensure differentiation capacity and functional characterization of the cell, to really make sure that the master cell bank that you have is acceptable and can be used in the future as a supply for the starting material. Because this in the end is what it is. It's a starting material for your off-the-shelf iPSC product. Also, during the process of the iPSC differentiation, which can take up to 60 days, even more if you think about some ophthalmological cell types. What is really important is to establish black and white QC controls very, very early on during the differentiation process, because these processes are really time intense and cost intense. So if you think about deploying a facility and all the machinery that is part of that facility, you want to make sure that as early as possible, you can essentially stop a differentiation run to save time, to initiate the facility with a new run, and to save money, not to continue a manufacturing run until you realize it's too late. So all of this is part of our general thinking and workflow, and that's what we are essentially working towards with our partners. I said genetic integrity is an important factor, and this is part of our standard testing for genetic integrity. And as you can see on the slide, on this table to the right, karyotyping is of course a method of choice, but we believe even in combination with arrays, CGH or WGS, this is not sufficiently covering the genetic integrity of the cell, because we made really some bad experiences in the past, not looking into enough methodologies. So we are really combining, in particular when it comes to key decision points, can we approve a master cell bank, an engineered master cell bank? We are combining different types of methodologies. On the one hand side, really unbiased genome-wide screenings, and on the other side, also really targeted methods where we are really looking into specific genes, into specific aberrations of the cell that are unwanted, to make sure that the cells are appropriately manufactured. All of this obviously is backed not only by wet labs, but also by a massive bioinformatics platform that we are running in the background. And this is just one slide really showing you the necessity for the genetic integrity. On the other side, I also said we are not only working with generic methods, we also have our proprietary methodologies. This is one example. We have patents on hypoimmune platforms, so to really prevent the rejection of the iPS cells that are finally transplanted to the patients. And I don't want to go into every detail of this, but obviously you have to consider NK cells, T cells, inflammation, macrophages, response to the cell types that you are providing. And I think it's very important to say that there is not one solution for everything. So we truly believe that for every different indication, for every different cell type that you are developing, an individualized approach is needed. But this individualized approach has some really underlying fundamental joint strategies that we believe we have patented in our proprietary hypoimmune platform. Finally, this is a slide summarizing what we have available. So many of you may know we are in collaborations with Sanofi in diabetes, and there will be a presentation tomorrow afternoon. I invite everybody to get an update on this as part of the scientific slam on diabetes. We have a quite interesting process development collaboration with Novo. Process development means we are not geared towards a specific indication, geared towards a specific cell type. We are working with them here, pan-indicational, really on technological aspects of differentiating cells. And our own proprietary portfolio really comprises different cell types. So we are working on genetically modified islet-like clusters for diabetes. We have available differentiation protocols for T cells, gamma delta in particular. We have a strong franchise in macrophages, and deploying this across different indications with a specific focus at this moment in time on metabolic diseases. So with this, I'm going to close this session, and in case there are any questions, because I know we are a little bit ahead of schedule, I'm happy to answer questions.